Hi, and welcome to Chapter 17 of Nucleic Acids. In Nucleic Acids, we're going to learn the story of how nucleic acids and proteins come together and are able to actually produce the proteins themselves. As we know, proteins are responsible for really everything that we do in our body. So everything that we are, everything that we do structurally, our enzymatic interactions, are all based on our populations of proteins. And nucleic acids hold or store the information for making those proteins within their bonds. Basically, nucleic acids are composed of either ribonucleic acid or deoxyribonucleic acid, and they're quite large, the molecules themselves, and they have monomers of the nucleic acids that fit together to make, well, actually monomers of nucleotides that fit together to make the nucleic acid. The nucleic acid monomer is composed of what we call nucleotides. And that is a phosphate, okay, that is connected to the 5' carbon of a sugar, okay. The sugar has a pentose ring. That means it has 5 members within the ring. And on the other side, on the 1' carbon, okay, on the first carbon, there is a nitrogenous base that is connected there. Okay, so there's 3 major components of a nucleic acid or nucleotide within the monomer. That is pentose sugar, a base, and a phosphate. The bases themselves are pretty easy to remember. For the final exam, I don't expect you to be able to draw each one of the bases, but I do expect you to be able to recognize, if you're told that it's a base, you need to recognize whether it's a pyrimidine or a purine. Just remember that the pyrimidine has a longer name, but a smaller structure. So it will be 1 nitrogenous ring, not 2 fused rings. The purines, on the other hand, a little shorter name. The purines will have 2 fused rings, okay? The pyrimidines that we have are cytosine, thymine, and uracil. Take a quick look at the thymine and the uracil. You can see that they're almost the same structure. The only difference is that there's a methyl group on the thymine that's not present on the uracil. We only find the thymine in DNA, and the uracil is the complementary base pairing of adenine in RNA. So why the difference? I imagine because methyls within the stored DNA serve as marker points, just like if you got lost in a forest and you were to put little flags on the trees. That's sort of what methyl groups serve as a way to be able to find things in the DNA. And therefore, it's not necessary once the nucleic acids become mobile. And the RNA is much more mobile form of the nucleic acids. So you do need to know that the pyrimidines include cytosine, thymine, and uracil, and the purines include adenine and guanine. And by the way, when we describe them, typically you won't describe them as their full name, cytosine or thymine or uracil. Rather, you'll just use the capitalized form, the capitalized letter of the first letter of the word. So cytosine would be base C, thymine would be base T, and uracil would be base U. And when we describe the DNA and the RNA, the different sequences that take place, we describe them with the base itself. So if you have a cytosine as your first nucleotide within your nucleic acid, then you would write C. And then if you had a guanine that followed, it would be G. If it had an adenine that followed, it would be A, and so on and so forth. Keep in mind that the cytosine, guanine, adenine are all the same in the RNA and DNA, but the thymine does lose its methyl group when it switches to the complementary base pairing in RNA, and therefore it's called uracil. Let's take a quick look at the pentose sugars. There is a distinct difference between the pentose sugars of a DNA and an RNA. In DNA, we can see that if we look at the 2' carbon, there's a hydroxyl group, and that is not present in the DNA. So RNA has a hydroxyl group where DNA does not. And that hydroxyl group is important. It actually serves as a means for that molecule to be broken down. We know that alcohols can participate in many biological reactions, and therefore when we don't have the alcohol present, it's not as easy to metabolize that molecule. And that makes sense. When we have DNA, we want to store the information. We don't want it to be marred. We want it to stay pristine. We don't want anything to be metabolized. And therefore, but when we have RNA, which is a mobile form of nucleic acid, we want to be able to control it. So we need to be able to metabolize it at will. And so that gives us the opportunity, sort of a chomping on point, to be able to break down the molecule if necessary. Now, a nucleoside is slightly different than a nucleotide in just its structure. Rather than having 3 basic parts, as we described with the nucleotide, a nucleoside only has 2. It will just be a combination of a sugar and a base. And it's linked with the sugar with a glycosidic bond at the carbon 1 of the sugar. Okay, so we have here at carbon 1, we get, it's actually called an N-glycosidic bond because rather than having an oxygen that's attaching it, it's going to actually have a nitrogen attaching to that point, and you'll lose a water as the reaction goes forward. So the N-glycosidic bond is slightly different than the ester bond in that it has a nitrogen that connects to the carbon rather than an oxygen. Okay, and this is just showing a ribose combining with an adenine to make adenosine. When you have nucleosides, you call them, well, we'll talk about the names in just a second. A nucleotide, on the other hand, going back to that second slide, a nucleotide has that phosphate group on the 5' carbon, okay, and it also has the glycosidic bond on the N-glycosidic bond on the first carbon. Okay, whenever you have a nucleotide present, you identify where the phosphate is in that particular nucleotide. If the phosphate is on the 5' carbon, you're going to identify that that's where it is. Okay, so in the first example, it shows you the RNA version of each one of these combinations, and then below it in pink, if we were to take off the hydroxyl group on the second carbon there, then it gives you the name that would be have that hydroxy group removed and the pink hydrogen put on carbon 2. Okay, so in the first example, we have adenosine 5' monophosphate, or AMP. Does that sound familiar to anybody? It should sound roughly familiar to ATP, adenosine triphosphate, and that's where on the 5' carbon you have 3 phosphates in a row attached to that. And we utilize that for energy. And so if you're seeing some similarities between this and biology, you should. Okay, so you can take a look at these, spend some time looking at them and see how they're naming them. But you notice that when we have a purine present, that the ending of the base name is going to end in SINE. And when we have a pyrimidine present, it's going to end in DINE. In table 17.1, it identifies what all the bases are in the DNA and the RNA, okay, the differences between the sugar and the ribose, and what a nucleoside is compared to a nucleotide. And that when we could describe them as nucleic acids, then we're talking about being a polymer. That's many monomers of nucleotides. Okay, so polymers, you can also have polymers of DNA and you can have polymers of RNA. Okay, so nucleosides will contain a purine or a pyrimidine, and your endings will either end with SINE if it is a purine, and IDINE if it's a pyrimidine. Remember to add the deoxy at the beginning of all the names if DNA is the type of sugar that is present within the nucleoside. And then if you have a phosphate present, you need to put the corresponding place or the carbon attachment where that phosphate occurs. Again, once we get into the nucleic acid, we start abbreviating the sequences by just identifying the bases, since all of the rest of the molecule is pretty much the same. It's the same phosphate backbone and nucleic acid, so on and so forth. In table 17.2, it identifies all the bases and how we describe the nucleosides and the nucleotides that are within those. Remember that nucleotides have the phosphates added to it. So a nucleic acid is a polymer, okay, of the different nucleotides or nucleosides, depending on what we're looking at, okay? And they'll have a unique sequence as far as where the bases are actually placed. It could be AAA, GGG, CCC, that kind of thing, or you could have something completely different. And as the bases alter in in their sequence that will actually translate in the end to a protein that has different amino acids. The primary structure of a nucleic acid is how the nucleotides are joined together. In other words, the base sequence that we see. Okay. Now, if we were to look at the molecule itself and kind of look at the different parts, we have to be able to recognize where the phosphodiester bond is. And the phosphodiester bond occurs typically at the 5' carbon of the sugar. Okay. So it attaches at the 5' carbon. We call the end that has the phosphate attached to the 5' carbon the free 5' phosphate. Okay, and when they start describing DNA from 5' to 3', they're saying there's a free phosphate at the 5' end and there's a free hydroxyl at the 3' end. Okay, and then of course we can see between the two sugars that there's a phosphodiester. There's two ester groups that are attached to a phosphorus. Okay, that are each attached to carbons on either side. So we call it a 3 to 5' phosphodiester bond. So the primary structure, again, of the nucleic acid is where are those, when we go from the 5' end to the 3' end, where are those bases? So in this case, we have an A at the top at the 5' end, then a C, then a G, then a T. Okay? So when we look here, there's a mistake on the slide. I just realized from Pearson. They wrote T on there, and it's really a U on the explanation. That should say a T at the 5' end in the last when they talk about the nucleic acid sequence of the bases represented by this. Now, if we were talking about RNA, it would definitely be a U, but they were talking about DNA here, so that's a mistake in the slide. In the primary structure of RNA, for this one, it will be from 5' end to 3' end, A, C, G, U. So it looks like they accidentally forgot to move the last comment to the next slide. Now, DNA itself is not just one linear strand of the primary structure. It has a secondary structure that involves two strands of DNA that are linked together with intermolecular forces. Specifically, the intermolecular forces are called hydrogen bonds. Okay? And we find that the A base or the adenine base will always link up to a thymine base or uracil. Okay, depending on if we're in if we're in DNA to DNA, it will be an A to T attachment between the hydrogen bonding. But if we are in RNA, DNA to RNA or RNA to RNA, then it will be an A to U attachment. Okay, but when we talk about guanine and cytosine, those will also have a hydrogen bond attachment between the two strands. So those are not covalent bonds, but hydrogen bonds between the strands. And when we have the A to T, you'll find that the hydrogen bonding will only occur between the carbonyl and the amines, or the amine and the nitrogenous base portion of it. And so there's only two hydrogen bonds between an A to T or A to U. But between a G to C, we have 3 hydrogen bonds. So it's a little bit stronger between a GC pairing than an AT pairing. So hence we get what's called the double helix. It's called a double helix because as the hydrogen bonds come together between the bases, the actual molecule, because of the intermolecular forces, will start to twist around itself just automatically, just due to the intermolecular forces. And so that makes a helical structure. Just like we talked about the alpha helices, we've got the double helix that is very, very similar to that. And so you get this sort of intermingling of the two strands. We see that at the top of the purple strands there's one that says 5' and one that says 3', because what will happen on the 3' is it will be flipped, the sugar will be flipped upside down, okay, so that it has the 3' at the top and the 5' at the bottom. And you can see that it's got the base pairing A to T, C to G. You can see that the little dots represent the hydrogen bonds that are taking place, which are just intermolecular interactions. They are not covalent bonds. So that means that we can separate the DNA from the two strands from one another if necessary. It's not a terribly strong bond, but in large numbers, it's very significant. Okay? DNA is responsible for 2 jobs, 2. And one is to replicate itself, and one is to be able to transcribe and translate the genes into proteins. First, we'll talk about DNA replication. Within all of your cells, you have DNA that's been replicated from its parent cells. Okay, whenever you have cell growth, or if an organism is growing and needs multiple cells to be made, then you have DNA replication going on. And that's just where the DNA unwinds, it makes an exact copy of itself for the daughter cell, and then during cell division, the DNA is then split up so that you get a copy in each of the cells. In DNA replication, the DNA is exactly like itself. It's not the same as the DNA replication for meiosis, where a baby's going to be made, but I was just talking about general DNA replication. When babies are made, then the DNA is switched around and you don't get an exact copy as you do when you have cell damage repair and things like that, where cells are just dividing and making daughter cells. So again, in DNA replication, the two strands will separate from one another. We see the original strands as the purple strands on this slide, and the blue strands are the new DNA strands that are being copied from the original strands. DNA fingerprinting is a method for us to be able to compare DNA from one source to DNA from another source. What we know about everyone's DNA is that it's all unique in the sequencing, in the way that the bases are placed next to each other. So the primary structure of DNA is unique to every individual. And for that reason, if we can't really work with a full strand of DNA. The DNA is too long and complicated, and quite frankly, DNA is sort of like snot. It's very gooey and hard to work with. So you have to cut it into manageable pieces to be able to sort of work with it. And so we use enzymes that will cut between certain specific base sequences, and we'll get certain lengths of the DNA that are exactly the same. If you do that in two separate samples and do the experiment separately and use the same enzyme, if they're the same DNA, you can put them on a gel that will separate out the strands by size, okay, where the smallest go fastest and are furthest along, and the largest bands are a little slower going through the gels. When we do that, if the DNA is from the same source, the bands that travel will be the same bands. And so we get this picture, if we dye the DNA within the gels, we get a picture of where the bands can travel to. And if they're the same, okay, as our control sample, then it's the same DNA. RNA, on the other hand, is a more mobile form of nucleic acid. And most of the nucleic acids are made up of RNA because we're using them to actually produce the protein. It's able to transcribe the gene from the DNA. So you're probably asking yourself, what is a gene? A gene is a stretch of the DNA found within the nucleus for eukaryotes, okay, that contains the directions to make every single protein that's expressed in your body. Anytime that you're put under unusual circumstances, new proteins will be expressed. If a new protein needs to be expressed, there'll be a cell signaling molecule that gets triggered on the outside of the cell. There'll be a cascade of reactions that then occur to send a signal to open up the stretch of DNA that contains the gene. And then a message will be transcribed that is made up of the RNA nucleic acid. Okay, and then the RNA then is, as it's transcribed, which means it's sort of like the original DNA message or the DNA gene is transcribed by the RNA. In doing that, going from DNA to RNA, we have to note that the bases will change from T's to U's. Also, whenever you have a message, okay, let's see if we get here. Let's get to the message. Okay, so whenever we're going to have protein being synthesized, okay, we're going to have a message transcribed within the nucleus right next to the DNA where the gene has been opened up, okay? Once it's opened up, a polymerase, okay, will attach to the DNA, and as it does, it will bring, okay, the nucleotides that correspond with that given base that's found on the DNA. So the polymerase runs along the DNA and it pulls down the appropriate base that is its complement. Okay, so the DNA template strand, that's the original DNA, will be attached to by a polymerase that will run along that original strand of DNA, and as it hits each base, it will pull the complementary base from its environment, the complementary nucleotide. So if you had an A, it's going to pull its complement, which is a U. If it has a T, it's going to pull its complement, which is an A. If it has a G, it's going to pull its complement, which is a C. And if it's C, it's going to pull its complement with a G. Okay? So A's from DNA go to U's from RNA. T's from DNA go to A's from RNA. G's from DNA go to C's for the message in RNA. And C's from DNA go to G's for the message in RNA. So the complementary strand is made. It's not able to just take a chunk of the DNA and then carry it over and then translate that into a protein. It's not able to do that. It has to do every single time that a process goes forward with protein synthesis, it copies it corresponds to the complementary base pairing. Okay, so let's go back again. In the nucleus, the DNA is opened up and a gene stretch that is responsible for the recipe for a particular protein, okay, is opened up. A polymerase attaches to the gene and it transcribes, writes down, the message. The message is the complementary base pair to the original DNA. The complementary base pair to the original DNA. Okay? Once that message is produced, it actually has to be protected. Once the message is produced, the next step is placing a guanine cap on before the methionine, which is the very first codon, and we'll talk about codons in a second. Before the methionine, there's a G cap placed on the front of it, and there's an A tail placed on the back of it. And A tail means a series of adenines that are placed after the last codon. Okay? Within the message, every pertinent codon that's going to be translated later into the protein, okay, will happen every 3 bases within the strand. So the very first one will always be methionine, and there'll be a codon for that. Every 3 bases will be a codon. Then the next 3 will be a new codon. Then the next 3 will be a new codon. Codons are only found on the messenger RNA. The messenger RNA carries the complementary bases to the original DNA strand. Once the message is transcribed, that means written down, and then protected, the message is then carried out of the nucleus and over to the endoplasmic reticulum. The endoplasmic reticulum in the body, let me go back to a better, these slides are a little mixed up for me. The endoplasmic reticulum is outside of the nucleus, okay? And in the endoplasmic reticulum, there's going to be rRNA or ribosomal RNA. That's also made up of RNA in combination with proteins. All the RNAs have a little bit of proteins with them except for the message, okay? So the rRNA will have a two-part subpart system. And it'll have a large part and a small part. And the message will read through those 2 parts. And as the message reads through, the ribosomal RNA will kind of clomp on to the codon. That means every 3 base pairs. So the first codon it will clomp onto, which will be inevitably it will be a methionine. Okay? As it recognizes what the codon is, it will pull transfer RNAs from its environment. The transfer RNAs will already be equipped with the bottom portion of the transfer RNA having the anti-codon. The anti-codon is the complementary base pairing to the codon. The complement, so if you had an A, the complement would be a U. If you had a G, the complement would be a C, and so on and so forth. Okay? So the anti-codon is the complement to the message, and the message is the complement to the original DNA strand. Now, the transfer RNA, on the top portion of it, you can see in the top of this picture, the transfer RNA will have that anticodon on the bottom that matches up with the base pairing from the nucleic acid. But on the top of the transfer RNA, it will have its corresponding amino acid that goes along with that codon, or anticodon rather. Okay, so when you look at it, then the transfer RNA is brought in, it binds into the active site, okay, and as it binds in, shifts, and is ejected, it will drop off an amino acid that will make a peptide bond. We know that's an amide bond between it and the previous amino acid. Okay, so the whole story. You transcribe the message in the DNA at the gene with the complementary base pairing of a message. That's produced by pulling the nucleotides from its environment and placing them in a complementary way to what the original strand was. The strand is then protected, carried out of the nucleus to the endoplasmic reticulum, which is another part of the cell that contains ribosomes. The ribosomes will be embedded in the endoplasmic reticulum's membrane, and they'll also be free-floating. But the only job is to act like a Pac-Man. It chomps down on the message. The message runs right through the ribosomal unit, and it will chomp down on a codon. As it does and recognizes what the codon is, it will pull the anticodon that's on a particular transfer RNA that will also have its corresponding amino acid, and as it clomps on, it will drop off its amino acid. As it ejects, the peptide bond will form between it and the previous amino acid that was dropped off on from the previous codon. Okay? So let's go back and review slower. Transcription is the first step in protein synthesis, and that's where the message is formed by doing the complementary base pairing. Okay? So G's will go to C's, A's will go to U's, A's will go to U's, C's will go to G's in this example, and the T will go to an A. Okay? During transcription, it is initiated with RNA polymerase. If you took a further class that's more complicated, it of course will have a lot more steps that you have to learn, but this is just the basics. Okay? After the message is transcribed, it's carried out of the nucleus to the endoplasmic reticulum, where the codons will be translated at the ribosomal unit into the language of amino acids. How do we know which ones are which? Some poor grad student somewhere along the way has tested every single combination of codons that are possible for 20 amino acids. So what we found was that there is a genetic code for the amino acids in which each amino acid can have a series of different combinations to make it. So each one of these combinations that you see on this chart, okay, code for one of the 20 amino acids. You'll notice that several of the amino acids have more than one combination that can make them, okay? They almost always have the same first letter and second letter for that matter. They almost always have the first two letters the same, but the last letter seems to be any single base. Okay. For instance, when we look at leucine, which is kind of second on the list, or third amino acid down on the list under U and C, and across from C, we see that leucine can be have a codon on the message of CUU, CUC, CUA, or CUG. So they're all the same in the first two bases in the codon, but the last codon is kind of whatever base is there. Okay, so for these combinations, there's going to be 4 different transcription RNAs that will be present floating around in the environment that will have the anticodon, the complement to these particular combinations. Okay, and if CUU is the codon, then the anticodon on the transfer RNA will be GAA. And on the top of that transfer RNA will be a leucine that eventually gets dropped off. Okay? So to determine the amino acids, you would have to follow the codon. The codon, of course, is the complementary bases of the original DNA strand. Okay, and you will get this table 17.4 on the final exam. So make sure that you understand how to use this and that it comes from the message, not from the original DNA, in order to get it translated in the endoplasmic reticulum with the transfer RNAs. Okay, so the initiation of protein synthesis starts at the message. After it's brought out of the nucleus, it's going to attach to the ribosomal RNA, and then it will always be initiated with a start codon. The start codon in the message is always AUG. That codes for methionine. That'll always be in every single protein, the very first codon that is translated. Okay, and that codon, of course, is going to have a complementary base pairing. Of course, that's with hydrogen bonds, as with all base pairing, to the transfer RNA for methionine in this situation. Okay, and then as the second codon attaches to with its transfer RNA, each transfer RNA will be unique. Okay, it will drop off the second amino acid. As those two nucleic acid, or sorry, not nucleic acids, but transfer RNAs are ejected from the ribosomal unit, then a peptide bond will form. Okay, the peptide bond, of course, is an amide bond. And here you can see the actual transfer RNA. At the bottom of the transfer RNA is the anticodon that's actually in the transfer RNA, and then it shows below it the hydrogen bonding that went to a codon on a message. Okay, at the top of the transfer RNA is a serine that's attached to the 3' end of that transfer RNA. Okay. Translocation just means that there's been a shift from the codon to the new transfer RNA, and so that just means that it's shifting. Okay, and so that's just a vocabulary word that tells you about the process. So as we translocate from one transfer RNA to the next one in the next codon sequence, okay, then the peptide bond will form, and then the next transfer RNA comes in, and then the next one's ejected, and then the next one comes in, so on and so forth. So that's translocation. Okay, the termination step will have a stop codon that codes for that. There's only 3 stop codons within all the combinations of codons, and that's UGA, UAA, or UAG. Okay, so what will you need to know for the final exam? I expect you to know everything about protein synthesis. So I expect you to know how to draw the complementary base pairing for the message, how to draw the complementary base pairing for the, which the message, of course, has the codons in it, and how to recognize how to put an anticodon on there. And if you have the codon's message, what it will translate to using the genetic code. Okay, so once we get the polypeptide forming its initial or primary structure, of course, then we've already talked about the different levels of protein. Once it gets its primary structure, it will start forming its secondary structures of alpha helixes, beta sheets, and of course, those will cross-link, form salt bridges and disulfide bonds, and all of the things that have effects on tertiary and quaternary structures. That's all from chapter 16. You can go back and review it if you don't understand what's going on. You can go ahead and practice these. This just says if you were to assign which concept would you assign to each of these descriptions. Ribosomes move along the message and add amino acids to a growing peptide chain. That, of course, is what? Translocating. You're moving the transfer RNAs along the ribosomes. If you are completing a peptide chain and it's released, you're terminating it. If a transfer RNA is attaching with its specific amino acid, we call it activation. And as the transfer RNA binds into the message, binds into the codon of the message, then we call it initiation. Okay, this one is just describing each one of those processes so that it's in a classified list. You can use this for the homework. That's table 17.5. 17.6 just tells us which type of nucleic acid we're working with and what its complementary base strand would be for each one of the processes along the steps of protein synthesis. We do have to keep in mind that every once in a while we have problems with the DNA where we have mutations take place. Mutations can be a series of things. It can be damage to the DNA. It could also be where some of the DNA was not copied correctly, so the wrong DNA base has been put in. So rather than having a C in one place, you have a G or something like that. Okay. So the result of having these mutations or mutagens, mutagens are actual components that cause the mutations. So the results of having the mutagens affect the DNA could be either substitution or frame shift problems with the DNA. Okay, and that will produce incorrect codon sequences to further be able to synthesize the actual protein. Okay, so that means that the protein is not being effectively made. It may have the wrong amino acids in the chain. It may even have a stop codon put on where it wasn't supposed to be, and the protein itself will be truncated or cut in half or cut at some point where the protein isn't completely formed. And then when the protein tries to make its normal shape and do its normal function, it's not able to. And typically with defective proteins, okay, and enzymes, which are also proteins, what will happen if the protein is non-functional is the body will make them because they've gotten a signal to make them. And if the protein is not produced, then the body's looking for the protein and it's not there, it doesn't recognize that it's there, then the body tells the DNA, we need to make more, we need to make more. And all of these defective proteins will build up and aggregate, okay, and make these masses, okay, and then you end up with disease, okay? Disease like galactosemia, cystic fibrosis, Down syndrome, muscular dystrophy, Huntington's disease, sickle cell anemia, hemophilia. All of these are aggregate diseases as a result of improper protein synthesis due to mutations. Okay, the types of mutations that can take place fit into two major categories. If you go into a later class, you might find that there's some categories that I didn't mention, but in this class, we're just going to mention the two basic ones. And that is a substitution or a frameshift mutation. A substitution is where at one point, at one base, you have substituted a different base in the original DNA. Okay, or there's been a substitution where the DNA is not read correctly and a different base is put in for the complement in the codon. One or the other, doesn't really matter. Each one of those would have a substituted codon where the base should be a different base than what it is. Okay, in that case, the codon frame is still the same, but your actual base is incorrect. Now, if it's the first two bases that stay the same and the third base is changed, oftentimes that won't matter, because remember that we have that wobbly base that I described when we looked at the genetic code. Let's look back at the genetic code. Okay, when we look at the genetic code of most of them, the third base that's in the codon is sort of wobbly. You can have a U or a C in phenylalanine, or you can have a U, an A, or a G in the third base in leucine. Okay? Therefore, when we look at this, you can have a combination and still get the same amino acid. In that case, when we look at the mutation, we would call that a silent mutation because it really wouldn't result in anything happening. Now, if you have a different amino acid sequence, okay, and there's been a direct change in the amino acid, that oftentimes won't matter either. If you substitute a nonpolar amino acid for another nonpolar amino acid, then you might not change the protein at all. It just might be a strange mutation and it really has no effect. The protein is still functional. If you have, let's say, a base amino acid or a basic amino acid, substitute for an acidic amino acid, then you may have some really distinct changes. It may cause the protein to fold in that spot, and so you get a malformed protein. All kinds of different things can happen. So it depends on where the substitution or the point mutation takes place. If nothing happens and we get the same amino acid sequence, we call it a silent mutation. A frameshift mutation is where you've actually taken out of the original DNA or the message, you don't have one of the bases, or you have put in an extra base. And then your codon shifts. Your codon frame is different. So every 3, now if you were missing one of them, then the first base of the next codon then is in the first codon. So you get a frame shift problem, and then when it tries to translate everything down in the endoplasmic reticulum, going from the nucleic acid to the amino acid sequence, then you're going to have a problem. It's going to be a completely new amino acid sequence all the way down from where that frame shift mutation took place. So that can be a very serious change. So again, the normal DNA sequence will produce a messenger RNA that contains a codon with the correct series of bases to translate with the transfer RNA into the amino acid sequence. Okay, with a mutation, here, if for instance, is a substitution, where we've substituted a C with a T, we've now changed the first base of the codon, and where it was a, let's see, glycine, where it was in the previous slide, a glycine, okay, now it will be a serine, okay, and that will be a problem, okay, so you have to kind of check and see what's happening. With a frameshift mutation, one of the bases has been removed. So in this case, we went from an original strand of methionine, lysine, phenylalanine, glycine, okay? And then when we have the frameshift mutation, we went to methionine, lysine, and then it went to leucine, and all the amino acids after that are going to be different, okay? So frameshifts are pretty serious. The effects of mutation, sometimes no effect. Other times, you have genetic diseases. What can cause these types of mutations? X-rays, UV sunlight, mutagens, which is chemicals that can genetically change how the DNA is behaving or covalently bonding, and viruses can also affect it. Okay, so examples, again, you can go back and check these diseases. We also need to just discuss very quickly viruses. Viruses are just mentioned in this chapter because viruses are composed of nucleic acids. Viruses are basically nucleic acids that have been encapsulated by a protein coat. And they are not really considered to be alive because they cannot regenerate themselves. So what viruses do is the protein acts as an injector, almost like a syringe, and it will inject the DNA or RNA into the cell and then utilize the cell's factory-like production of proteins to be able to produce more of the virus. And then once the virus becomes in an environment where it's precarious for the virus, then it will produce many, many viruses and cause the cell to burst, and then it will spread the viruses to new cells. Okay, so that's all we really need to talk about today. These are some of the diseases that are caused by viruses. You can go ahead and check them out. I will not be asking you much about viruses on the final exam, but I may mention it a little bit in the review if there's a couple of questions that might refer to viruses being nucleic acid sequences that are injected into the cell. Okay, so in this chapter we talked about nucleic acids, okay, which are composed of either DNA, deoxyribonucleic acid, or ribonucleic acid. Specifically, that nomenclature tells us that the sugars are different in the second carbon. DNA is missing its hydroxyl group on that second carbon. Okay. The other difference, of course, is in the bases. We have demethylated versions of thiamine. So our base count in DNA is going to be A, T, G's, and C's, whereas in our nucleotides, it's A, U, G, and C's. And our base pairing in DNA is going to be A's to T's and G's to C's. Our base pairing in RNA or RNA base pairing with DNA, either one of those, is going to be A's to U's and G's to C's. Okay, and when we look at nucleic acids, if we're talking about DNA, will be arranged in a double helix as a nucleic acid polymer. Okay, but with RNA, it's slightly different. This is another major difference between RNA and DNA. RNA is found single-stranded, not double-stranded with the complementary base pairing. The only time that it double strands up is when it's performing one of its complementary actions, where it is complementary pairing up with the original DNA message, where the codon it has a complementary pairing up with the anticodon, so on and so forth. We utilize the genetic code to be able to translate what is happening from the world of the nucleic acids to the world of the amino acids. Remember, the genetic code is talking about the sequences on the message, which is the complement to the original DNA. So on the message, you just find your codons and then you can translate it using the genetic code. You will get exactly the same chart that's found in the book on your final exam, so you don't have to memorize any of these things. Okay? So protein synthesis is a result of the DNA and RNA working together after being signaled to open up a gene. Okay? And that's the end of that talk. And I hope you guys have a great day. Make sure that you go to the final exam module and review the final exam. Make sure that you go to Facebook and log into the final exam review and participate in that so that you learn it as we go along. Have a great day.